Although in earlier sections we were talking about appearance of osteoblast cells and osteocytes as we were trying to introduce the concept of compact and spongy bone tissue, it is now time for us to finally demystify the process within a human body, within a human embryo and fetus, which essentially forms the bones. It is important that we understand that there are two different paths that bones will have to go through. One is a bit of a shortcut, and this is what we call the intramembranous ossification process, which means that there is a membrane that is made of mesenchyme that directly becomes converted into the bone. And then a little bit longer process that is called the endochondral ossification, through which the mesenchyme first forms the cartilaginous model of a bone, and then the cartilaginous model becomes gradually replaced by bone tissue and practically at one point starts looking like a bone. Uh, Two different processes are not going to have approximate 50-50 split within the body. Only few bones would need to go through shorter process of intramembranous ossification, and it is relatively easy to remember which bones. First group of bones that will ossify through the intramembranous ossification are bones that form the skull cap, because most children are born in a natural form and their heads are going to be the first leading part through the birth canal. Therefore, in order to protect the brain, we're going to need to have bones of the skull cap already forming a fairly firm shield that will protect the brain during the labor itself. Then we're going to have the lower jaw bone, the mandible, which also undergoes the same fast track process because the newborn, as we know, need to have lower jaw being quite solid, quite movable in order to support the basic things that the newborn will have to do in order to support the feeding and later on also to help a newborn to start vocalizing. The rest is just one single bone, the clavicle and the collarbone, which needs to be there as early as possible. That's why it goes through the intramembranous ossification, because it is the only bone which connects axial skeleton with skeleton of the upper limb, appendicular skeleton. So the clavicle is there practically to support this connection of axial to appendicular skeleton. Let's take a look now at some of the images that we have available, and let's talk a little bit about forming of the bone through both intramembranous as well as endochondral ossification. This is process of endochondral ossification illustrated in 5 different segments or 5 different sequences. So you know that it's going to be mesenchyme that forms the cartilage. So for a little while, instead of skeleton, we're going to have the cartilaginous models that are very similar to what the final product's going to look like. And you can see it has diaphysis or shaft, and then it has two ends or proximal and distal epiphysis. But the cartilage is a tissue that is given plenty of nutrients, and as a result, some of the chondrocytes, cells that make the cartilaginous tissue, will be consuming way more than what they need for their metabolism, and in their attempt to grow, they will essentially start bursting, practically destroying themselves, generating part of the cartilaginous model where no more living cells is found. That becomes the opportunistic moment for newly developed osteoblast cells that will keep invading the area. And as they do so, it is going to be also done with a lots of blood supply that is coming to support them. So the process of ossification will start within the center of diaphysis or body or shaft. That is what we call the primary ossification center. And then the process of ossification will start continuously spreading towards proximal and distal epiphysis. So on the diagram 3, you can see here that diaphysis is nearly completely ossified, but the same process still has to happen and to affect both proximal and distal epiphysis. So with the appearance of blood vessels and more nutrients that they're going to brought into the area, we're starting the same process of forming, this time just secondary ossification center that's going to be seen within proximal and distal epiphysis. So the secondary ossification center starts from the inside and moves outwards, as illustrated on diagrams number 4 and number 5. As the process advances towards diaphysis, it is just a question of time when this tiny strip of cartilage will be overrun by advancing bone. On a diagram 5, it is still illustrated like a very thin cartilaginous strip. That is metaphysis that we said earlier. And this will enable bone to increase its length. But towards the end of puberty, of course, the bone tissue advancing from diaphysis as well as advancing from the epiphysis direction will close this gap and it will turn it into epiphyseal plate. On this diagram, we can see also in 4 short segments what is the process that forms the bones of the skull cap, the mandible, and the clavicle. Therefore, we're talking about intramembranous ossification. The skull cap is made of mesenchyme, and at one point, as we know, the mesenchyme will progress and deliver first osteoblast cells into the area. That's going to form the primary center of ossification. It is illustrated on part of the diagram number 1 as these 5 osteoblast cells have produced this white colored halo around them, that is the matrix, that will subsequently calcify, and of course these bones will be trapped, these bone cells will be trapped in their own calcified product. They will become osteocytes and they will continue residing in their own lacunae. But in the meantime, new osteoblasts become laid on the surface of the primary ossification center, and it drives it outward, something that we can compare to a snowball effect. But as you can see, in vicinity of this primary ossification center, we already have 2 blood vessels, and we're not to see them being wiped away. Quite the opposite. These blood vessels will be incorporated into ossification center, which keeps expanding. The value of blood vessel is not necessarily to bring more nutrients to osteoblasts, but also to start bringing osteoclast cells. So here is a blood vessel that is still entering what looks for a moment as a bone made of just compact bone tissue, but with activities of osteoclast. On the diagram 4, you can see that the central part, the core part of the bone, will be converted into spongy bone tissue, and that is something that precisely fits into description what we said earlier for the bones of the skull cap. They fit into category of flat bones, which means that they're made of 2 layers of compact bone tissue, but the core part of the bone is generated by spongy bone tissue.